Hey everyone, let's wrap up Chapter 10 by having a short discussion on what prokaryotes do when they need to divide. So everything that we talked about in the preceding 3 videos, talking about the cell cycle, interphase, mitosis, and cell cycle regulation, really just applied to multicellular eukaryotes. So what we're going to talk about here is something that is actually going to apply towards prokaryotic cells. So there's going to be some similarities in what they try to accomplish as they go through the cell division process, but as you can probably imagine, with these being much simpler organisms, the process is going to actually be a lot simpler as well. So a couple of important distinctions to make here, especially when we're talking about prokaryotes. Prokaryotes tend to just have one circular chromosome. It's not a linear chromosome, it is a circular chromosome, and that is actually going to lead to a little bit of a complication we're going to have to deal with here in just a couple of minutes. So prokaryotes tend to have a single circular chromosome, so we're not having to go through all these elaborate processes to duplicate a bunch of chromosomes, then make sure all the right chromosomes end up in the daughter cells. We've only got one chromosome. We need to duplicate it. We need to make sure the original goes to one daughter cell and the copy goes to the other daughter cell. You'll recall that the chromosome here is not contained within a nucleus because prokaryotes do not have nuclei, but rather it's folded up in an area in the center of the cell called the nucleoid region. A prokaryote still has the same sort of compaction problem that we talked about before. In the case, the DNA is actually about 1,000 times the length of the cell. So some association with histone-like proteins is actually necessary to compact the DNA so that it's all going to fit. When DNA replication occurs, this is actually going to form 2 rings of DNA, and we're actually going to see that these 2 rings are interlinked with one another, so we're actually going to have to find a way to unlink them and separate them before the cell division process can actually occur. In order to ensure that both chromosomes end up where they're going where they're supposed to go, since we do not have spindles here, we do not have microtubules in the sense of how we understand microtubules so far. Since we don't have that elaborate mechanism for pulling the chromosomes to where they need to go, the way that we're actually going to fix that problem is we will actually attach these replicated DNA rings to the plasma membrane itself. So you'll see what we mean by that here in just a minute. So the process of splitting the cell in 2 in this regard, it's not called cytokinesis, it's actually called binary fission. So you're going to replicate your chromosome, and so you'll have 2 replicate chromosomes. You'll distribute both of them to the 2 daughter cells, as we said before. One of the major things to understand here is that this is asexual reproduction. What we mean by that is that since this is a process in which we are dealing with single-cell prokaryotes, each individual cell is an individual organism. So since this binary fission process produces a new cell, it produces a new organism, and it is a type of reproduction. To give you some context for the time scale of this, not every type of prokaryote is going to be exactly the same, but E. coli, which is a very common bacteria that we deal with on a daily basis, it has a division time of about 20 minutes. To give you an idea of what exactly this means, if you have 1 million E. coli bacteria, 20 minutes later, you're going to have 2 million. If you wait another 20 minutes, you're going to have 4 million. If you wait another 20 minutes, you're going to have 8 million. So every time 20 minutes passes, the number of bacteria doubles. So this whole binary fission process can happen very, very, very quickly. Do you remember how long the cell cycle, the entire cell cycle takes for a human cell? Yeah, it's about 24 hours. So prokaryotic cell division is much more simple and can be accomplished in a much smaller amount of time, namely because there's a lot less oversight that needs to happen. We don't need to spend a whole lot of time preparing and checking and everything like that. Okay, so here is a depiction of the binary fission process. If you look at the bacterial chromosome, there's a very particular part of the chromosome at which DNA replication is going to start, and this is called the origin of replication. So once we duplicate the single ring circular ring-like chromosome, we're going to have two ring-like chromosomes that are actually bound together. So later, we are going to have an enzyme come in and break the phosphodiester bond of one of those DNA molecules so that they can be unlinked, and then we can stick them back together. So the two chromosomes will then be attached to the plasma membrane near those origin of replication. So you can see here, in this case, this chromosome is stuck to the plasma membrane here, and this one is stuck to the plasma membrane here. We do this intentionally so that we can make sure that these two chromosomes end up attached to the plasma membrane as far apart from each other as they possibly can be, and this maximizes our chances of making sure that the two daughter cells end up with one chromosome each. Now, another thing as far as the binary fission process go, I said that we do not call this cytokinesis because we do not use a cleavage furo. We do not use actin, we do not use myosin. Instead, the bacteria are going to use a tubulin-like protein called FTSZ. So this is a very closely related protein. It very much resembles tubulin, but it's not tubulin. So what this FTSZ protein is going to do is start directing the formation of a membranous septum in the space in between the two nucleoid regions. It's very much like how we make a cell plate in plant cell cytokinesis. So what this is going to do is form a membrane in between the two cells to pinch the two cells off from each other. And then if we're talking about a prokaryote that has a cell wall, we can synthesize a cell wall in the space in between the two septums. So to kind of look at some big picture stuff here, if we want to compare binary fission with mitosis, which we're a little bit more familiar with at this point, they are similar in the sense that both of them are going to generate genetically identical daughter cells. However, mitosis does not generate new individual organisms if you're talking about a multicellular eukaryote. It does if you're talking about a single-celled eukaryote like budding yeast or something like that, but if it is a multicellular eukaryote, mitosis does not produce new organisms, so it is not a case of asexual reproduction. So if you're talking about multicellular organisms like us, mitosis is not for reproduction. It is just for growth, renewal, and repair, as we have said several times already. But binary fission, what prokaryotes do, is the method by which they reproduce. That's asexual reproduction. So since each daughter cell receives one copy of the parent cell chromosome, each cell is going to have the same chromosome, but each cell will be its own unique, individually functioning organism that then they can spread out and possibly obtain mutations or new plasmid DNAs to become unique in their own right. So if you look at this table here, you can see a little bit of a comparing and contrasting of cell division and in prokaryotes and cell division in eukaryotes. So everything that we've understood up until now, we talked about cell division in multicellular eukaryotes, in which case we use this for development, growth, and repair. In other cases, if you're talking about single-celled organisms, it is actually for asexual reproduction. So if you want to talk about how reproduction is going to happen in a multicellular eukaryote, mitosis is not what you want. As we said before, the problem with mitosis is that you maintain your chromosome number. So you start with a diploid mother cell and you end up with two diploid daughter cells. So if that were the case, then every generation after ours would have twice as many chromosomes as the ones that came before it if sperm and egg both had 46 chromosomes. So we in the next chapter are going to explore with meiosis how meiosis generates haploid gamete cells, the sperm and the egg, and that becomes our method by which new organisms can be created and conceived. All right, that's going to do it for Chapter 10. Join us next time and we will start talking about Chapter 11 and meiosis and sexual reproduction.